Today, we have discussed one of the important bacterial disease, that is the angular leaf spot of cotton. In this angular leaf spot of cotton, we have discussed about what is the pathogen, what is the host, and what are the symptoms we are observed on the plant surfaces, even though which part is more affected by this disease, and how to the disease management controlling disease control methods, that is integrated pest management methods, and the biological control or cultural methods and chemical antibiotic methods we are all discussed yesterday class. So today we are going to discuss about one of the important other bacterial disease, that is what we can say that bacterial blights of paddy. Okay, this slide is visible. Bacterial blight of paddy.
Yes, sir.
So in this bacterial blight of paddy, we are going to discuss about what are the symptoms we are observed in plants and how to identification of this pathogen, and what are the management strategies for the controlling of this disease, and along with that disease, which type of cultural methods we are observed. Along with cultural methods, what are the chemical methods we are going to adopt in this control methods of these plant diseases. So today we are going to discuss about the bacterial blights. Okay, bacterial blight of paddy. So we know that in our India is the agricultural country, you know. So more than 65 population, they depends upon the agriculture sector. So agriculture is one of the dominant sector in all over India, which is employed more than 65% of the people. They are depends upon agriculture, directly or indirectly. Okay, so in the agriculture sector, one of the most important crop, that is our day-to-day life, we are using this paddy. That means rice. Am I right? So rice is one of the most important grain or most important cereal, you know. So so that's why we are going to discuss about the rice. That the rice affect different pathogens, whether may belongs to bacterial or whether they are belongs to the viral pathogen. We are going to discuss. So in this topic, we are going to discuss about the bacterial blight. The pathogen belongs to bacteria, and what are the symptoms we are observed in this bacterial blight. What are the causal organism of this bacterial blight? So the causal organism of bacterial blight of paddy, that is Xanthomonas oryzae, plant variety oryzae. So we know that. What is the scientific name of our paddy? Anybody can tell? Oryza sativa, you know. Oryza sativa indica. So Oryza sativa is affected by these bacteria. That disease we can call as bacterial blight or bacterial leaf blight of paddy. So the symptoms we are going to discuss here. So first, seedling or crest and leaf blights, and the confirmation whether the disease belongs to seedling wilt or leaf blights. Okay, so we are observed 1 to 3 weeks transplanting of our paddy. Then the green water soaked layer along the cut portion of the leaf tip of leaves as early symptoms. We are observed on the surface of the leaves. The leaves then going to wilt and roll up and become grayish green to yellow color. And afterwards, the entire plant is going to wilt completely. The complete plant going to wilt. So in this leaf blight, the water-soaked yellowish strips on leaf blades or leaf lamina. Leaf blades also we can call as leaf lamina, or the starting at leaf tips with a wavy margins. Wavy margins. Then afterwards, the leaves with undulated yellowish white or golden yellow coloration we are observed on the leaves surfaces. And the marginal necrosis. Necrosis, we know that yesterday we are discussed, you know, necrosis is nothing but the tissue is going to die and decaying. So this marginal necrosis and dying of leaves back from tip and curling, and the leaving midrib intact are the major symptoms we are observed. Then this appearance of bacterial ooze that looks like a milky or opaque dewdrop on young lesions early in the morning. So whenever the severe affecting of this disease in severe affected, the leaves tend to dry quickly. So in this seedling wilt or crest, the entire plant will going to completely wilt. At the same time, leaf blight, we are absorbed, the severe infection absorbed, then automatically the leaves are going to tend dry quickly. And loss grain yield may up to 60%. So 60% of the loss we are absorbed. And confirmation, how to confirmation whether this disease belongs to bacterial or whether the disease belongs to viral disease, or if the bacteria, that is a leaf blight disease or other disease. So the distinguish Kresge symptoms from stem borer damage. The lower end of the infected seedling can be squeezed between the fingers. Then yellowish bacterial ooze may be seen coming out the cut ends. The cut portion can be absorbed against the light to see the bacterial ooze streaming out from the cut ends into the water. After 1 to 2 hours, the water becomes turbid. Then the infection of the pathogen. So what is the pathogen, my dear friends? That is Xanthomonas oryzae, variety oryzae. So this infection, the bacterial causing the disease are rod-shaped bacteria. So they are rod-shaped bacteria, and that is 1.2 into 0.3 to 0.5 micrometer range diameter we are observed. The pathogen are single. They are occasionally pairs, but not in chains. So single means bacillus, bacillus. They are not paired, okay, but they are not chains. They are gram-negative bacteria, gram-negative bacteria. So yesterday we are discussed, angular leaf spot cotton, also gram-negative bacteria. And non-spore forming, they are non-spore forming bacteria. In yesterday, the angular leaf spot cotton also non-spore forming bacteria. Okay, and the colonies nutrients agar are pale yellow, circular, and smooth with entire margin. They are convex in viscid. Whenever we are going to observe in the laboratory method. So in this laboratory, these are the in Petri plates we are observed. These are the structures we are observed in Petri. Then what are the factors favoring for this disease development? So any disease, the favorable conditions is most important. So the favorable condition is especially in this bacterial blight, the presence of weed. So the weeds are the major problem for the spreading of this disease, and presence of rice stubbles and ratoons of infected plants. Even though the presence of the bacteria in the rice paddy and irrigation canals, and the temperature 25 to 30 degrees, that means tropical region and subtropical region, entire world is going to affect this bacterial blight. The high humidity. So yesterday, so 86% of the humidity we are observed in angular leaf spot cotton. And even the rain and deep water is also suitable for the transmitting of this transmitting of this bacterial disease. That is a bacterial blight. And severe winds. So wind species also cause the wounds and the over fertilization also. So whenever we are using nitrogen, ammonia, and superphosphate like fertilizers, the excess of the fertilizer, whenever we are using, then this disease spread very rapidly. So these are the suitable conditions for the development of the disease. Even though the irrigation water and splashing wind blown rain can also suitable for the disease, transmission of this disease. And what are the preventing methods we are adopted for the controlling of this disease? So in integrated pest management or even the disease management system, we have to use seed treatment. So yesterday we are also discussed, seed treatment with bleaching powder. See here, so this is the bleaching powder. So while the seed treatment, we must use the bleaching powder for 100 gram per liter. Even though along with this seed treatment, zinc sulfate 2% reduces the bacterial blight. Are you follow my class, my dear friends? Anybody? Anybody one can rest?
Yes, sir.
Yes. Then at the same time, seed treatment or seed soaking for 8 hours, agromycin. So agromycin. So in this slide, we are image showing this agromycin 343. And the vegetable ceresan, followed by hot water treatment is also used for prevention of this disease. At the same time, seed soaking for 8 hours, then ceresan 0.1% and treat with streptomycin. Streptocycline is an antibiotic, 3 gram in 1 liter we should use. At the same time, along with this, spray neem oil 3% or NSKE 5%. See here, this is the neem oil. Are you ever heard neem oil? Vepa nune.
Yes, sir.
Neem oil. Neem oil is one of the best pesticide, biopesticide, So pesticide from extracted from plants, so which is very useful and also very eco-friendly pest. So by spraying this neem oil 3%, it is control the bacterial disease. And one more important, even though the cow dung, pashula paid antama, telusu kada? So this cow dung is also, the fresh cow dung is also suitable for the control of this disease. Which disease? Bacterial blight. So how to use this fresh cattle dung or cow dung? By the usage of this cow dung, dissolve 20 gram cow dung in 1 liter of water and allow it settle into sieve and use a supernatant liquid, and this used for the treatment of the this disease. And some of the cultural methods also we are used. So some of the varieties, disease resistance varieties, disease resistance varieties. What is that? What are they? IR20,
IR20, IR72,
very good.
Ponmani,
and
TKM6.
TKM6. Very good. So these are the best suitable varieties for the tolerate for this disease, resistant to the disease. So bacterial blight is controlled by these varieties, IR20, IR72, and Ponmani TKM6. These are the varieties when we are used without using any pesticides. And at the same time, secure disease-free seeds. Whenever we should protect or preserve a disease-free environment and grow nourishes and preferably in isolated upload conditions. And avoid clipping of seedlings during transplanting. So whenever we are going to transplant, should be avoid clipping or seedlings and previous seedlings. So that is the most important point. At the same time, we are should be used with the suggestion of the agriculture officers. That is a balanced fertilization, So avoid excess of the nitrogen application. Whenever we are using urea, so urea is nothing but it is contain the nitrogen, So nitrogen content is more than other nutrients. So that's why whenever we are applicable the nitrogen fertilizer without take the suggestion from the agriculture officer, it is very harmful to the crops. So that's why should be skip the fertilizer whenever we are observed this disease among our crop. So should be avoid nitrogen content fertilizers. And even though drain the field except at flowering stage of the crop. Should be, my dear friends, our crop field needs more than 60 to 70% of the water every day. Should be change the water, or sometimes should be drain in the field is the most important. So whenever water is going to flow from our crop field, that is very important, and also it is avoid the problem of this disease. And should be separate from the croplands, weeds. So should be separate from our croplands. Avoid flow of the water from affected fields. And my dear friends, remember, the affected fields is also suitable for the transmission of this bacteria from affected field to the healthy croplands. And even though maintain proper plant spacing, should be spaces. See here, among the lines of our paddy field, the space is also very, very important for the control of this disease. And other culture come from.
Sir, I have doubt in last page.
Yeah.
Sir, what is avoid clipping of seedling during transplanting?
Yes, my dear friends, have you ever seen our how to cultivate this paddy in your very nearest croplands or agriculture field? Anybody? So generally, we are transplanting of this paddy. We are just the seedlings. We are what we can say. మనం ఏం చేస్తామంటే వాటిని ఒక్క దగ్గర అలుకుతారు అనమాట రైతులు. ఒక్క దగ్గర అలికినటువంటి వాటిని పెరిగిన తర్వాత సెపరేట్ చేసి ఏం చేస్తారంటే ట్రాన్స్ప్లాంట్ చేస్తారు, మళ్ళీ నాటుతారు వేరే దగ్గర, నాటు వేస్తారంట. మనం అర్థమవుతుందా?
Yes, sir.
సో అలాంటి వేసేటువంటి సమయంలో ఇన్ ప్రీవియస్ వెనెవర్ వీఆర్ ట్రాన్స్ప్లాంటింగ్ ఆఫ్ దిస్ క్రాప్స్ ఆర్ ద ప్యాడీ బై ది ఇన్ ప్రీవియస్ సీడ్లింగ్స్ ఆర్ ఎనీ అదర్ clippings there na should be separate or should be removed from our crop field. Why? Because that is also cause for the transmission of this disease. Okay.
Sir, what is clipping, sir?
Clipping, avoid clipping means the previous crop field, previous crop. For example, in our paddy field is suitable for continuously cultivating paddy only. So in previous year or previous season we are already cultivated paddy. So whenever we are harvesting our crop, the ప్యాడీ వాట్ వీ కెన్ సే దట్ మన ముందున్నటువంటి ఆ కిందున్న మొలకలు అదేవిధంగా గడ్డి అలాగే ఉంటుంది, తెలుసా మీకు? వాటిని మనం ఏం చేయాలో తీసేయాలి. ఇక్కడ చూడండి, అర్థమవుతుందా? పాతవి మనం రిమూవ్ చేయాలన్నమాట. అర్థమవుతుందా? అర్థం కాలే మీకు? ఇంకా మొక్కలు ఈ ఆరోగ్యవంతమైనటువంటి మనం ఏవైతే మనం వేస్తున్నప్పుడు పాతగా ఉన్నవి లేదా వ్యాధిగ్రస్తమైనటువంటి వాటికి సంబంధించిన క్లిప్పింగ్స్ కానీ చిన్న చిన్న కట్ చేసినటువంటి ప్రీవియస్ బయట ఉన్నా వాటిని ఏం చేయాలి? సెపరేట్ చేసేయాలి, రిమూవ్ చేసేయాలన్నమాట. షుడ్ బి రిమూవ్.
We want to remove all the stubbles, sir.
What we can say?
Stubbles.
Yeah. Then the chemical methods. So along with this cultural methods, chemical methods is also used for the controlling of this disease. Okay. So seed treatment with bleaching powder. At the same time, seed treatment with agromycin. Okay. And seed soaking 8 hours treat with streptomycin and spray streptomycin sulfate. Streptomycin is an antibiotic, Streptomycin is an antibiotic. At the same time, application of the bleaching powder 5 kg per hectare in the irrigation land. Okay, and water is recommended the crest stage. Okay, and even though the foliar spray with the copper fungicide alternated with the streptocycline. Okay, so these are the chemical methods we are using for the control of this disease. Okay, so this is about the bacterial blight disease. If you have any doubt, please ask me. So now, if you don't have any doubt, we will move for the viral diseases. Any doubt undamma? Any doubt? Madam ji? Any doubt?
No, sir.
Okay, Madam ji unnara?